Okay, good morning, everybody. We're continuing discussion the regulation mechanism with respect to translation. And for that, as you remember, we left the last lecture on the elongation steps during the translation process. And I break down all those steps from the recognition of the start codon up to the association of the next tRNA, so the entry to the A site, the peptide bond formation, which is due to the peptide transferase center, which is part of the large subunit of the ribosome, the translocation, so there's sliding of the ribosome over the mRNA, and this sliding is of 3 nucleotides, which correspond to the codons. At this point, this sliding will remove the previous tRNA will exit from the E site, and the cycle can start again, okay? When we speak about peptide bond formation, that means it is with respect to the tRNA, which is named the peptidyl tRNA because it is the one that is covalently bound to the chain. And the new one, which is the amino acid tRNA, which is the one bringing the new amino acid. And the reaction is between the carboxy terminal group of the peptidic chain, and it is a transfer from the interaction with the tRNA, which was a covalent binding to the tRNA. So this one has to be hydrolyzed, and the formation of a new condensation reaction, which would be in between this carboxy terminal part, carboxylic group, and the amine group of the new amino acid that at this point will be part of the chain. So it is a dehydration reaction. Through this reaction, water molecule is formed, okay? And the final result is that now the chain is associated to this tRNA, which was the one that was bringing the new amino acid. And the empty one is not, will slide at the exit site and will leave the interaction with the ribosome. Okay, that's all steps. So for this reason, the first amino acid will always be the N terminal, and the last one will correspond to the C terminal. Okay? And after elongation, when we reach termination, the termination will means the hydrolysis of the covalent binding in between the chain and the tRNA without any other addition, and that's the reason why the chain will be released. So this is, okay, the summary of those, but let me discuss on this diagram, which is the termination phase. What happens at termination? So you remember that the termination site will be close to the polyadenylation signal, meaning the polyadenylation signal signals also that we are close to the termination. And the termination corresponds to a codon that doesn't code for any tRNA. There's no anti-codon. There are no tRNA with a complementary sequence with respect to the stop codon. And more precisely speaking, we shouldn't call those stop codon, but it would be more correct to speak about stop signals. Because when we speak about codons, usually it is because of the presence of an anti-codon. So, but it's so usual that we just speak about stop codons. But remember, there's no corresponding tRNA for those. Instead, there are termination factors that will interact with those stop signals. So the stop factors are with this type of name, which are the E stands for eukaryote. That was the same thing also for the initiation and the elongation. So the E is just to differentiate the mechanism for eukaryotes and the regulation of eukaryotes ribosome with respect to prokaryotes. So prokaryotes have owner names and thus slight differences, okay? So the E is only for eukaryotes. And RF stands for releasing factor. So those are the factors that will allow the release of the chain, meaning they interact when the ribosome is reaching those stop signals. You know, there are 3 stop signals, and they are all binding site for those releasing factors. And guess what? Which molecule will we find as regulators of this process? Small g proteins. So don't be confused with the name. The ERF1 is the protein interacting with the stop signals. The G protein is ERF3. And just to be sure we don't get confused, just I added GTP. Okay, so since it is bound to GTP, this one is a G protein. And the interesting part is both will interact until the association of the ERF1 is stably associated to the stop signal. Why that? Because there might be an association that would be a wrong one. It is because those stop codons are not that different from the coding one. So there might be an association which is with two base pairing, but not with a third one. And that's not correct. It shouldn't be there, let's say, in other words. Because don't think that the things will come right away with the best, or let's say the right tRNA directly associating with the codon. You have to figure out that everything is a matter of those molecules, and they try to interact, okay? And it is the right interaction that is stabilized. Just to be sure, we are not interrupting the chain wrongly in a wrong manner, because the interaction of this releasing factor would be pretty good, but not the exactly one. So the interaction with the G protein is here for the accuracy of the interaction. And only when there's a strong, stable interaction in between the releasing factor 1 with the 3 nucleotides coding as a stop signal, when the interaction is stable, it is possible for the elongation, the releasing factor 1 to behave as a gap for the releasing factor 3. So the releasing factor 1 has several roles. One, the specificity for the interaction with the stop signal. Second, the activity, that is the capacity to activate the GTPase activity of the releasing factor 3 by this process, because the interaction changed somehow the organization of the factor 1 in the configuration in which it's stably interacting with the DNA. This promotes its gap activity towards the releasing factor 3. At this point, the releasing factor 3 will activate the GTPase activity. And by hydrolysis of the GTP to GDP, now it will move away from the releasing factor 1. Okay? So it's a little bit tricky because it is the same protein that has the role for the specificity of interaction with the DNA sequence and also the gap activity. And that's not the only activity of this protein. So by moving away from the interaction with EF1, EF3 release allow for the next step. And the next step will be the accommodation of other proteins. But at this point, I think there was no need to add some more names of the molecules. The important point is this will favor the hydrolysis by the peptidyl center, the hydrolysis of the chain, and the release of the chain. Okay? So here, I've lost meanwhile the numbers in the conversion and in PowerPoint and Keynote. Okay, this was the number one. This one is the number two. So at first there was a recognition, the interaction with RF3 with RF1. At this point, RF1 will function as a gap where due to the GTPase activity of the RF3, this one will deactivate itself by hydrolysis of the GTP, and this one will promote the release of the RF3 from the interaction with F1. Okay, so this was number three. Here we are in number four. That's okay. This one is a way allowing for the activation of the peptide transferase center that will promote hydrolysis, as it does, because that's also what happens in the formation of the peptide bond, because we have first to release the interaction with the tRNA. So this first part happens, but since there's no new amino acids to add, because there was no amino acid there, okay, because there was no binding of tRNA, at this point, the chain is released. That's okay? And all this will promote the detachment, not only the release of the chain, but also the release of the interaction in between the small subunit, ribosomal subunit, and the large ribosomal subunit, and the release of the interaction with the mRNA. At this point, the small subunits can start again the interaction with a new mRNA, and the cycle will start again from initiation to elongation to termination. And most probably, the same small ribosomal subunit will associate with another mRNA, maybe not the same one, will be read it again by this subunit. It will be by others. And this small subunit interacting with 5' mRNA will bind up to the start codon, and at this point we'll associate a large subunit, which will be probably different from that one. Okay? That's the reason why the ribosome by themselves do not exist unless they are busy translating an mRNA. Is that okay? Okay, I thought a diagram of that was useful because it's quite confusing. The interaction on one hand, the activation, mostly when it is the same protein that will do both activities. Speaking about learning, the important part is pick up the whole regulation, okay? And if you don't remember if it's the RF1 or RF3, the number by itself at this stage is not the important one. If that helps you, use the numbers and remember the right name of those proteins. But also during the exam, if you happen to not remember if it is the one or the three, I prefer if you don't remember the number, but you pick up how it works. Okay? That's more important. So just to be clear, don't bother too much about all those names. We'll give you by the end a list of the ones that you really should know, because those are the ones that you will find recurrently, okay? While many of those, if you happen to work maybe on a disease which involves the regulation of those molecules, you will learn of those and also of others participating to those regulation. But that means you will be more specialist of this type of regulation. Okay, you cannot be a specialist of each time of regulation. Nobody is. No way. It's so difficult. Okay, now let's speak about numbers, because up to now we didn't spoke more about numbers. So which are the numbers? 20, 45, 61. That tells you something? 20 amino acids. 45, 61 codon. 45 is a number of types of tRNA, at least in higher eukaryotes. So it can change. It is 45 to 50, and some are very rare one. So let's speak about 45, just to pick up a number, okay? So what do you have of that? The standard amino acid. That means without modification, not the rare one, so on, just the standard one. So that the one we'll find in the genetic code, okay, tables. You know that they are divided according to the characteristic of the residues, since on the other hand, they share in common the carboxylic group and the amine group, which is the one for the skeleton of the protein. Okay, those are the ones that are involved in the formation of the peptide group. So I don't want to go through the whole discussion. I just to point out three of those, because those ones are definitely important, since they are the ones that can be phosphorylated. There are three nucleotides mostly. It's not exclusive, but three that are the one you will hear the most, and that's serine, threonine, and tyrosine. And that's because of those hydroxyl group in their residues, okay? And those hydroxyl group will allow for phosphorylation. And as I already mentioned, and you will hear more and more about those regulation through phosphorylation. That means in the peptide chain, each time there's a serine, a threonine, a tyrosine, those could be targets for the enzymes take care of the phosphorylation, and those are named kinase. So the kinase will interact with those amino acids and transfer from ATP, the phosphate group, to those amino acids. That's what we meant by phosphorylation. And since you had a phosphate group, that means that you had negative charges, and negative charges will mean changes in the structure of the protein. Okay, or it might be also a way for protein interaction because of the complementarity of the charge. Okay, so for this reason, those three, from our point of view, are the important one mostly, but that doesn't mean that the other one are not. But it's just from the point of view of signaling and post translational modification. Okay, what about the numbers? So we mentioned 20 amino acids, and there are 20 enzymes able to manage the covalent binding of amino acids to the tRNA. So those are the amino acid tRNA synthase, because it says what it does. It will allow the synthesis of the amino acid, let's say the amino acid, to the tRNA. Okay, so it has several pockets. With those cartoons, it's quite difficult to manage with everything, but just remind there will be pocket for the interaction of the tRNA with this protein, and mostly with the part corresponding to the anti-codon, because that's what gives the specificity mostly, and also with some of the amino acids that are surrounding the anti-codon. Okay, so that's the reason why you can see here the interaction in between the amino acid tRNA transferase and the proper tRNA. And on the other hand, the other pocket will allow for the interaction with the amino acid. And that makes sense that the interaction, because of the specificity part, is with the residue. Okay, so that's the residue that interacts with the pocket. And it leaves the carboxylic group close to the tRNA, because that's a carboxylic group of the amino acid that will be covalently bound to the 3' of the tRNA. Okay, so the 3' of the tRNA, because of the position in the interaction with this enzyme, is nearby the carboxylic group, and this enzyme will allow for peptidyl binding, the formation. Okay, I took the example, let's say if this one is specific for phenylalanine tRNA, this one will be a tRNA with an anti-codon that codes for phenylalanine because of the specificity of the anti-codon. The interaction is specific, and on the other hand, this pocket is specific for the amino acid phenylalanine. So what will be the end product? It will be the phenylalanine tRNA, meaning it is a tRNA that is loaded with its amino acid. So this one is very important, because if it is the wrong association, there is no way the ribosome can make any control over this error, right? Because it will be only a matter of interaction, anti-codon, codon, and what is the amino acid over there is not under the control of the peptidyl center. Okay, so the peptidyl transferase is the one that is very important in that case. Okay, so at this point, I just changed it graphically. Okay, so let's say this one is just now, it is the same thing. Okay, I just changed because that's where it was too difficult. It was impossible to see it. So this tRNA loaded with phenylalanine group amino acid, at this point will be used in the translation process according to the elongation process we just described. Okay, and in the exit site, it will be the same tRNA, which is not anymore loaded, but it will be ready for recycling, interacting again with its phenylalanine tRNA synthase. And that means that those tRNA cycle in between the the situation in which they are loaded with the amino acid, involved in the translation process, exit as unloaded, the free one, and the free one is ready to interact again with this protein. So there are only 20, but each of those are in high numbers because they have to take care of the loading of all the tRNA. And for this one, all the phenylalanine tRNA. And there's only one for all the phenyl. Okay, I should have chosen one that was easier to pronounce. Phenylalanine amino acid. Okay, so for this reason, we have the correspondence is 1-to-1. There are 20 amino acids and 20 amino acid tRNA synthase. Okay, but we have 61 codons. So since we have 61 codons, there are more than one codons coding for the same tRNA, for the same amino acid, sorry. Okay? There are two important things. The genetic codon, first of all, it is universal for all the eukaryotes. It's slightly different for prokaryotes, even if it works exactly in the same way, there are few differences. And when it comes really to the codon, okay? But in general terms, the genetic codon is universal because it works for both prokaryotes and eukaryotes in the same way. But the table is not exactly the same for prokaryotes and for eukaryotes. So it's unambiguous, meaning that each codon codes only for one amino acid. Okay? While one amino acid can be encoded by more than one codon. That's the redundancy aspect. Because of the fact that it is unambiguous, that means that by knowing the nucleotide sequence, you can unambiguously telling what is the amino acid sequence. Because to one codon, the correspondence is only with one amino acid. It doesn't work the other way around. Because looking at the sequence of a protein, for each amino acid, except for methionine, because methionine has only one codon, which is AUG. And methionine is the only one with only one codon. Okay, if you look at that, this one has only one. While for all the others, there are at least two. And in case of leucine, there are 6. Okay, that's the redundancy. So now, how can we manage with those numbers and the redundancy? How can we fit? So we answered somehow part of those, meaning in between the genetic code and the 20 amino acids, it's a correspondence which is unambiguous, but there's more than one codon coding for one amino acid, except methionine. So what happens with the amino acid synthase? The amino acid synthase we just mentioned, there are 20. So they have to manage with the redundancy. there are 45, let's say, in humans, it's between 45 and 50, but some are very rare ones. So let's speak about 45 types of tRNA. So we have 45 tRNA, but 61 codons. So it's not a one-to-one match, right? So how can all those match together? Which are the answer to this question? First of all, and it might be the easiest one. Okay, this one are the summary of those. So on one hand, there are all the production of the tRNA. On the other one, the nucleotide, the amino acid, sorry. Okay, so we have 45 of those, 20 of those, and 20 of the enzyme, the T amino acid synthase. Okay. We mentioned that there's one for each of those amino acids. So that means that they will accommodate 45 tRNA. That means that some of those will accommodate more than one tRNA. But what do they have in common? It will be the one that code for the same amino acid. In other words, the phenylalanine one that we mentioned before, if we look at the genetic code, you see we have two codons coding for phenylalanine. So one possibility is that the same enzyme will accommodate two types of phenylalanine tRNA, namely the one with the anticodon that is complementary to UUU and the one that is complementary to UUC. So this one as an anticodon will be AAA. This one as an anticodon will be AAG. And this is the case. So the first thing is that some of those amino acid tRNA synthase accommodate more than one type of tRNA. So if we use as an example, alanine, for example, if you look at the genetic code, alanine is coded by 4 codons. Those 4 codons have 2 nucleotides that are in common, and the third one can be of any of the 4 nucleotides. The redundancy is most of the time on this third product. By the way, the third product looking at the mRNA sequence is the first anti looking at the mRNA sequence because they are anti-parallel. Okay? So in this case, the 4 codons for alanine will interact with the same analeal tRNA synthase. And since the complementarity, the anticodon is not always the same one, there will be the possibility for this enzyme to accommodate very precisely in between the amino acids and the nucleotide, the complementarity in the structure, it's not hybridization, it is the interaction in between amino acids of the protein and the RNA part of the tRNA in the region of the anticodon, and the common part are the nucleotides that are surrounding this anticodon site. So let's say you see the anticodon are different with respect to the first nucleotide, okay? Because it is the last one on the codon. So from this reason, the complementarity in the recognition will depend on the surrounding amino acid. So those parts will be the same one. And those parts are the ones that will allow for the specific recognition of those four among the other tRNA. Okay? It is because of the surroundings of the other nucleotides close to the anticodon that allow for the interaction of all the four with the same amino acid tRNA synthase. So for this reason, because of those regions, those tRNA interact with this. Yes, this was the one. This alanine tRNA synthase and not with another one. If I go back there, okay, that's the reason why. Okay, too bad for me. I changed in another one and not updated. So you read it here, alanine. That's why the colors don't match. Okay, so this one will accommodate the four for alanine and not the other one. That's the reason why you keep the one-to-one specificity with respect to all the amino acids. But that's not enough because, okay, that explains part of the redundancy in between the number of amino acids and the number of codons. At this point, we understood how different tRNA coding for the same codon can interact with the same amino acid tRNA synthase. But that means 45 possibilities, not 61. So we are missing some tRNA for the interaction with some codons. You got lost? You have 20 enzymes able to connect tRNA with amino acids. There are 20, okay? You have 20 amino acids, so that's much. With respect to the pocket for the amino acid, this one is one-to-one, okay? On the other hand, you have a pocket for tRNA, okay? And the pocket for the tRNA is not one-to-one. It will be one-to-one for which one? Methionine. That's the only one. For the others, it might be with 2 or it might be with 4, okay? There's a possibility. So we can match. Now we understand how we can match 20 amino acid tRNA synthase with 45 different tRNA. That's okay? That's part now we can understand. That's much. But still, we have 45 tRNA. But in the table, there are 61 codons because the table is 64 minus 3, which are the stop signals. So that means 61. Now It's a matter of how can we solve that 45 tRNA allow for a genetic information with 61 codons. That define the universe, the fact that only those will be bound with this amino acid. And the accommodation at this point cannot be strictly with respect to those three. It is with two. And the last part is because of the surrounding. Okay? So the surrounding are important as well for the specificity of the recognition. That's okay? Okay. Now, we are missing those, let's say, 16 codons for which theoretically there's no, not theoretically, there are no tRNA corresponding one. And the explanation comes with what is called the Wobble hypothesis. What do we mean by Wobble hypothesis? This means that there is a kind of tolerance with respect to not to some unpairing. So look at this situation. In this situation, here are two tRNA, okay? Here are the 2, 1, and 2, and they are bound to the same amino acid. And this one is the case, for example, with phenylalanine. So in that case, with phenylalanine, there are two products, UUC and UU. And there is only one tRNA, which anti is UUG. So there's no anti-codon with the HAA. It doesn't exist. It is part of those missing 16 tRNA we were speaking about. Okay? And the Wobble hypothesis is that the matching will be okay with the two first nucleotide of the codon. So here they are complementary. With this anti-codon, it is complementary also with the third one, while this match will happen during protein synthesis, but due to the mechanism, it's allowed for this type of interaction that is with a guanine interacting with a uracil. So it is not the right one, the one that's the canonical one, okay? Because usually the interaction is in between C and G. But in that case, the C is in direct interaction with the G, the U with the A. But in the Wobble, in the third position, this interaction, and you see it will be a substitution of a pyrimidine 1 basis with the other one, which is also a pyrimidine. And the hydrogen bonds can form sufficiently for this to be stable enough to allow the peptidyl reaction. So the wobble mechanism means for the missing tRNA, or there are no tRNA with some of the anticodons, and the solution comes from this wobble hypothesis, meaning the interaction is stable enough to allow for the reaction with the peptidyl center. You see, here is phenylalanine, here is leucine. They are both with the two starting nucleotide of the codon, which are the same, UU. But both can accommodate not only the right complementary tRNA, but also the other one, in which there is a switch in between the purine bases or the pyrimidine one. Leucine is also encoded by 4 other codons that do not start with UU. Okay, when we looked at the table. So now we could understand how the match in between 20 amino acids, 20 amino acid tRNA synthase is possible with the 45 tRNA because of the accommodation in the pocket due to the same surrounding nucleotide with respect to the specificity of the amino acid. And for the others, the missing tRNA, let's say, it is because of this tolerance when there's a switch in between the two pyridine or purine bases, which means that the same tRNA binds to 2 codons on the 3rd position, and sometimes even higher. The same RNA, the tRNA is the same in that case. So that's exactly the situation, because it is the same tRNA. If you look at the anticodon, you see the anticodon? The anticodon is the same one. The anticodon is AAG. The other one is AAG. Okay, so this one is the same tRNA because we have only 45, and they interact with 2 codons. But your question was the contrary around? It can bind, usually it is with 2 because we have 2 purine bases and 2 pyrimidine one. And you save the type of interaction with the number also of hydrogen bonds. The other one will come from the other mechanism, in which for the others, you have different tRNA, okay, loaded with the same amino acid, and those ones take care of the other part of the table. Yeah, okay, because it is, by the end of the day, it is a match in between 20 amino acids and 61 codons. And we sum, one of the parts is because of the explanation of the 45 tRNA interaction with the same, each one with its specific amino acid tRNA synthase. And the last part is because of the wobble hypothesis. So at this point, we have the same tRNA that can interact with two codons. And that's the reason why in the table, we'll find that even if it is one codon or the other one, we end up with the same amino acid. That's also the reason why you have conservative mutation with respect to the protein sequence. Because in many situations, if you change, or if there is a mutation, or if there is, we have also our specific genetic information. Okay, so on one hand, it's specific to each of us. On the other hand, they code for the same types of proteins. That's the reason why you can analyze the fingerprint of each of us. But on the other hand, they all code for the same proteins. It will be a balance, it will be equilibrium, it will be regulation differences. Okay, that's the reason why we are not clones, we are different. So we manage with the differences, but with respect to the sequence of insulin protein, if we take all of us, we'll find roughly the same sequence. When we speak about the amino acid one, we might have differences with respect to the DNA sequence, just because if we change the third nucleotide of the genetic code, in that case, if someone has written in the genetic code UUU or UUC, that's a difference in the genetic sequence, but that makes no difference with respect to the protein. Okay? And that's also because of the unicity. When you read the sequence of the genetic level, okay, so some of those mutations have no consequences. That's only type of specific, let's say, characterization of the DNA, but it has no effect on the protein structure and the protein functioning and the protein activity and so on. So when you remember the sequence I showed you, I brought the real one, the real gel one, but I didn't mention when he did it, and the sequence was already published, but we wanted to manipulate, and at that time it was not available. We had to produce it in any case. And when you produce with recombinant DNA and all those cut and paste and reverse transcription and all the amplification and so on, by the end of the day, you have to check, okay, is that exactly the sequence I'm interested in? Is that exactly the right one? Because you can have errors in many of those steps. Okay, we are dealing with enzymes and so on. So okay, and when we did and we looked, it was okay. And at one point, one base reading the gel was not the correct one. At this point, we say, oh my goodness, because that might, some of those could be mutations that make the protein behaving differently, or you may lose even the frame, the reading frame, if you there's one nucleotide added. Okay, so very anxious. So and pair with the published one, and hopefully it was according to the frame, the reading frame. It was on the third basis of one of the codons. So at this point, we relaxed and said, okay, we can go ahead with this DNA that we had cloned, and that was okay. Okay. I was, that's what makes me smiling, because I was looking at some of those information on the first genome project, okay? And there was a kind of historical aspect and dimension. How was it in the lab and so on? And the picture was of someone reading exactly the same type of gel I showed you. And because at that time it has been done manually. And that's, it sounds nonsense today. It says, well, how was it possible? And there was a whole bunch of scientists working on that because at that time we had no way for automation, and it changes in 30 years in such a way that's really incredible. And it will also with you. That's the nice part of it, because we keep on learning so many of those things. The bad news for you, maybe for you, when I was a student, as you are, there was no cell biology, no molecular biology, no bioinformatics. There was nothing like this. It was only with genetics, biochemistry, physiology. That was the only thing. All those topics, we didn't know anything about it. That's because I keep on teaching elsewhere. That would have been so boring, if it would have not evolved and you have to repeat each time. But each year you just add new things because that's the very nice part of it. It's increasing and it's exponential. Okay, and the bad news for you is you have much more things to know about, but we try to manage in any case. Okay, so what's small g protein doing over there? I already mentioned it, so I'm not. This is just to make a kind of summary. This is the type of thing you might do to prepare your answering the questions to the exam, just to figure out, okay, this question wants me to reorganize information in a different way. So where can I pick up different parts just to figure out an answer that makes sense? So you can yourself reorganize the information, and trying also with some of those diagrams just to summarize and keep in mind, okay, from that one, I remember that this one will do this one will do the other one, okay? Okay, which are the mechanism of regulation? All those, the availability of the initiation factors. If you're running out in the supply of initiation factors, you will slow down translation. You can also make changes in the elongation part. If you have the interaction with microRNA, for example, those will impair the sliding of the ribosome. You might have to do with the regulation of those binding proteins in the process. And another aspect is the stability of the mRNA, because for translation, you need the mRNA. So one of the regulation is about the mRNA. The translation process can be also favored. You increase the efficiency by allowing for the initiation of ribosomes one after the other one. So that's what this picture is illustrating. You start from the 5 prime, okay? So the first 5 prime is the cap, it's on that side. So here is the AUG, and at this point, the ribosome attach the small subunit attached on the 5 prime, slide up to the AUG. At this point, the large one will sit on it, and we go ahead for elongation. So at this point, it was only with the AUG, the methionine, and we add the large subunit. And you see this growing peptide. Here it is at the beginning. This ribosome is prior to the other one. So the peptide is longer, and you go ahead, and this one is close to the stop codon and will release the peptide. And you can see in electron microscopy, if there's a chance to see it, it depends on the plane of the section, but this one is pretty nice. And you see this organization in which the center part will be with the 5 prime, and here is the 3 prime, okay? And it is like a factory. This mRNA is AVily translated with ribosomes that it's not the sequence of the same ribosome going on. It is at the same time, as you can see with this electron microscopy, it is at the same time many ribosomes that are busy translating this RNA molecule, okay? And you can see those both as free ribosomes in the cytosol, and it can be seen sometimes also at the level of the endoplasmic reticulum membrane. So that would increase translation, okay, maybe with respect to the amount of protein that is produced in the same time. The other way could be an inhibition of translation, and the inhibition, as we mentioned, could be because the initiation factors are not available, or there are some inhibitory proteins. So those ones, you cannot initiate new translation. And you know what? When the cell is under stress, meaning the things are not properly in equilibrium, it's, for example, the cell is accumulating proteins which are not properly folded. You remember the folding, the 3D organization? You need chaperones to help for the proper folding. So if you're running out in the number of chaperones, you're accumulating proteins that are not properly folded. Is that right? Is that okay? Accumulating proteins which are not properly folded? No. Why not? And how the cell can detect proteins which are not properly folded? With respect to hydrophilicity and hydrophobicity, what will do the folding? Where will be the hydrophilic amino acid on the surface of the protein? Where will be the hydrophobic? Inside the structure, the 3D structure of the protein. Improper folding means you exposed hydrophobic region on the outside. And what do the hydrophobic region? The hydrophobic region, what do? They clump together because of hydrophobic interaction. They like each other because they collaborate to exclude water molecules. Okay? So what do they form? Aggregates. That's the reason why you cannot tolerate as a cell the accumulation, if it is too much, of non-properly folded proteins. So if the reason for this unfolding of the protein is because you're running short in chaperones, what's the solution to solve this problem? I will never finish that program. Okay. But okay, next things. Okay, how can we solve? We're running short in chaperones. So first of all, you stop the translation process. You slow it down. Okay? How can you do that with what we've just said? What we need to start translation? Interaction at the which side of the mRNA? 5 prime. And we need this 5 prime to be available for the interaction. Okay. So you might have inhibitors of the interaction of the small subunits with the 5 prime. That's slow down translation. That's a good point. So we take that one. That's a good one. But is that enough? Because now that we slow down translation, what do? We stay, okay, stand by? No, we have to solve the situation for the translation process to start again and the proteins to be correctly folded. What do we need for the proteins to be correctly folded? Chaperone. Denaturation? Oh no, maybe you mean to get rid of the unfolded protein? That's what you're meaning? Yeah, that's one possibility. And that will happen, okay, when you are in balance. Okay, in the regular situation, it's not that every protein is folded perfectly. There's no way. In biology, there's no the perfection, the thing that it always works perfectly, it will be like this. It doesn't exist. Okay? It is the capacity to manage to identify where is an error and correct it. Okay? So you're right. This is another good one, but it is in regulatory basis that happen. But now we are speaking about, okay, to do what you're saying, you need chaperones and chaperonins because you have to unfold and refold again. But that's a regular, the day by day basis, okay? And you need for that chaperones. But you're running out of chaperones. That's the reason why this one is not enough, and we are accumulating unfolded content. What do we need for the proper folding or to do what you were saying? You will need which protein? Chaperone. How can we get chaperones? By producing them. How will we increase chaperone production? Regulation of transcription. So at this point, this will means activation of which class of proteins that will regulate transcription? Transcription factors. So the transcription factors that will bind to the responsive elements that we find in the regulatory region of the genes coding for chaperones. That's a kind of brainstorming, okay? So this process, we need sensors, meaning there are proteins that can be activated because of the production of unfolded proteins. Those will activate the transcription factors of the one that bind to the responsive element of the gene coding for chaperone. Okay. Everything is okay. We are transcribing. Now we have a whole bunch of mRNA coding for chaperones. What happens? Translation. But we've blotted. Translation will happen anyway, because elsewhere we would never recover from this situation. But the amazing thing is that in that case, the translation of the mRNA coding for stress proteins, namely some of those are chaperones, do not require the 5' to be free. The interaction of the small subunit will be directly with the mRNA without sliding from the 5 prime. So it is because of a different regulation of the translation of the mRNA that code for chaperones that you can slow down the translation machinery for all the other proteins, leaving the possibility for the translation of the mRNA that are coding for chaperones. Okay? So again, I come to your question. The general rule is that the small subunit interacts with the 5 prime and slide up to the APG and blah, blah. Okay? But there are exceptions, and the exceptions allow to respond to those stress situations in which it's necessary to slow down the whole machinery, but leave the possibility for the translation of some mRNA. And those mRNA have a different characteristic for the interaction with the small ribosomal subunits, allow to bypass the 5 prime cap inhibition. It is one of the regulatory processes that is not a general one. It is linked to the structure of those mRNA rather than it's just a subcategory of mRNA that can be translated without the need of the interaction with the 5 prime. So they have another interaction mechanism in between the nucleotide, the small subunits of the ribosome, and the mRNA. So the mRNA for chaperones, no, it is only for the stress molecules. It is only for the chaperones. We took the example of chaperones, but it's wider. It is the stress situation. And the stress situation with respect to folding, you need a specific way for those regulations. So at this point, we can recover from this situation because we are favoring the production of the chaperones. These chaperones can also maybe solve some situation of the one that were not properly folded. Elsewhere, we have mechanism for those aggregates to be digested by lysosomes, so we get rid of those, and we don't keep on accumulating unfolded protein because we slow down, balance again the number of chaperones molecule with respect to the needed. Okay, at this point, we can start again with if there are chaperones, no more accumulation of unfolded protein, no more inhibition of the 5 prime cap initiation. And you can start again. We are always in the situation of those regulation and the balance. Yeah, those usually are tinted, translated through this mechanism because it depends on the structure of the mRNA. I'm not excluding that some might be translated according to both systems because one doesn't mean it's excluded the other one, but that means that if the 5' is blocked, they can be translated in any case. It doesn't mean they are always more translated. It means that it will be also a matter of how much of the mRNA coding for the chaperones are available. It's not, the regulation is not only on one part. Before we could solve the problem, we had to increase the transcription level. So we increased the mRNA level and we can translate those because they're already chaperones. So it doesn't mean that before there was no chaperones. There were chaperones, but they were not in a sufficient quantity. If you don't mind, I finished 5 minutes earlier, but I don't stop by now. It sounds too short. And swear we have 10 minutes and that's not enough. So, okay, it's pretty early. You're recovering. Okay. So that means that you have proteins that can bind in the 5' UTR, the one that is not translated. You might have also those elements that when interact in the 3' UTR. You have the, okay, those are the different possibilities. Just for you to understand that it doesn't mean, okay, translation works like this and it works like this for every mRNA and it works like this for the speed of the translation, that is always the same one. What we discussed was a slowdown of the general translation machinery because we were accumulating unfolded proteins as a whole. If we speak about microRNA, those are specific for specific mRNA. So in that case, it is the inhibition of the translation of specific mRNA and not of the whole RNA machinery. Okay, pick up the difference. It's possible to slow down for the whole machinery, and that's what we discussed. It also, but currently, the production of proteins relay on transcription level, but you remember that's not the only way. We have all those regulatory RNA. We discussed splicing. We produce several types maybe of mRNA coding for related proteins. And when they are translated, we're another way of regulating what's happening, and that might be through the microRNA. And the microRNA, by binding specifically to mRNA, can slow down, reduce the translation on one mRNA, but not of the other one. Okay? So when it comes to microRNA, it's a matter of specificity. One type of RNA that is slowed down, or it can even lead to the degradation of those mRNA, so they're not anymore available for translation. So that's specific for a specific type of protein. And you can understand that if you change the regulation of the microRNA, you change the effect is on the amount of proteins you get. And if this protein is regulating some important behavior, you can also switch from physiology to pathology through the change at the level of the microRNA situation. Okay. And that's what we call the mRNA decay, meaning the mRNA are produced, okay, by the transcription. They are used, translation, but they will be also with, let's say, present for a certain amount of time in the cell. They are not stable forever. Well, how will be the decay? It will be by shortening the tail. The poly A tail is one of the aspects. As, when the ribosome is translated, another aspect is the activation of exonucleases that can remove nucleotides from the 5 prime. It can destroy the 5 prime cap. And if we lose the 5 prime cap, we do not translate anymore this mRNA. Okay, so all those are other way to regulate the lifetime of each mRNA. So it can be with the exonucleotic activity. That means it is an enzyme that removes nucleotide from the extremities, or it can be endonucleases. You know, the restriction endonuclease, they cut in the middle of the DNA sequence. In that case, those are RNA endonucleases. They cut the chain in the middle and not from the extremities. That's the difference in between endonucleotic decay and exonucleotic decay. Okay, the exo are from the extremities, the endo cut in the middle. It can be also a decay because with respect to stop codon. So the mRNA that code for a few amino acids and there's a stop, obviously there's something that was not correct there. Those one will be identified or gained by interaction of those regulatory processes and degraded. The same thing for the mRNA that are missing a stop codon. And the RNA that are missing a stop codon will be identified by the cells due to protein interaction and degraded. So you see there's a whole bunch of what we could say quality control. And finally, the location. So some of the mRNA can be sequestrated in interaction with proteins and not available for the interaction with the ribosomal subunit. So another way to decrease the quantity of mRNA and some types of mRNA might be not degradation, but interaction in clumps forming granules. And those granules are a way to slow down maybe the translation of a specific protein. And if this one is a very important one for regulatory processes, you have to switch it on, but also to switch off. And to switch off might be not only waiting until it is translated, because all the process, I didn't speak about energy, but all those processes, when we speak about the regulatory aspect, when we speak it is with GTP hydrolysis, and ATPA is also involved. So you need a whole bunch of energy to produce the protein. If you don't need it anymore, you are not waiting for these proteins to be produced and later you degrade the protein. It is better if right away you remove the mRNA. Okay, so there's no more translation over there, and you will go for degradation. So all those just for you to keep in mind that each step is under control. Okay. Each step can also be monitored if it is okay, and if it is not, there are solutions to stop or reduce, to correct, and to start again. There are solutions when something has been switched on and it needs to be reliably regulated and switched off. There are different ways to switch it off. A protein that is a tubulin, for example, so actin, probably will not be so sharply regulated. because it is one of the housekeeping genes, the one that in any case you need and so on. But if it is a transcription factor, do you think the mRNA for regulatory transcription factor will be there for a long time in the cell? No, because elsewhere you lose the capacity to switch on and off. So there will be a regulation on the activity of the protein because that's the fastest one. But there's also the regulation on the side of transcription and translation. That's the reason why we're interested in transcriptome and proteome, meaning the profile. How are those different proteins balanced? And what is the difference in between a healthy situation and maybe a pathologic one? Okay. Okay. Speak with respect to clinics, you probably already know it, but when it comes to antibiotics, the target of the antibiotics are the translation process, and the target are the ribosomes of prokaryotes. It has been, let's say, the evolution used those antibiotics that are produced by microorganisms just for the equilibrium in between microorganisms, okay? And we use it to our own benefit to fight against bacterial infection. So that's also, since the target are the ribosomes from prokaryotes have different characteristics with respect to the one from eukaryotes, okay? And that's the reason why those antibiotics will fight against the functioning of the prokaryote ribosomes and not the eukaryotes one. That's also the reason why antibiotics are not useful in viral infection. Why that? Which ribosomes are used in viral infection to produce a viral protein? The one from the cell. So it is not the target of antibiotics. And how do you feel when you take antibiotics? Do you feel very healthy? How do you feel? Sleepy, or let's say tired. Why that? Oh, you're speaking about our own prokaryotes. Yeah, we are made by much more prokaryotes than eukaryotic cells. Okay, in our own body. There are much more prokaryotic cells than eukaryotic cells. Keep in mind also that one. But yeah, that might be a side effect, let's say. That's part of it. But that's not the one that I was thinking about, speaking about ourselves. More generally, mitochondria. Why that? Because the ribosomes of the mitochondria are much closer to the one of the prokaryotes. And if the mitochondria is not working properly, what happens to us? We are running short in ATP. That makes quite simple. Okay, it's not that simple, but somehow you can connect those things together. Okay, make connection. How primary proteins become functional? It is a matter of folding, and that's with the example we made, what I explained you. And this is due to all those post-translational modifications. So it's not only a matter of primary sequence, also the secondary one, the tertiary one, the quaternary one, for the protein to be biologically active. Okay, so all those post-translational modification are essential for the biological activity of the protein. Those modifications will relay on the primary because if you can form an alpha helix, it is because the composition of the amino acid over there allow for the formation of an alpha helix structure. But it doesn't mean that the alpha helix structure will always form, because it might be a matter also of regulation through the interaction with the chaperones. So the chaperones are part of the organization of the 3D structure of the proteins. And it is also a matter of the other enzymes that will change some characteristic of the primary sequence. For example, if you form the sulfide bonds, that's a covalent binding inside the same chain that will contribute heavily to the 3D organization and the biological activity of that chain. So it has both aspects. One is due to this primary sequence, and the other is the environmental condition in which is this protein for its 3D organization. And finally, one important aspect is also the localization and the activation. And we already somehow discussed those, because the 3D organization is one aspect. The fact that the protein will be active biologically or not active will depend on interaction. We've seen it in the interaction with the GTP, for example, if we speak about GTPase protein. But it is also a matter of regulation and compartment, because we've seen that by changing compartments, the biological activity of the RAN protein would change. So the biological activity has to do with the structure, has to do with the covalent modification also of the protein. It has to do with protein interaction. It has to do with protein localization for the fully active protein. And here I made a mistake because I have to change, and I don't want you to move. I have to change and switch to the next presentation, but few slides I have really to go there and do it. So let me just opening that one. Not that one, because it was the same one. Oh, that's the one. Let's speak about protein targeting. So there are signals. For this discovery, because it's a very important one. And what are we speaking about? It is about the sequence of amino acids, which has to do with the localization of the proteins. We already discussed those two, not speaking directly about the sequence, but speaking about the nuclear localization sequence. That means import, and the nuclear export sequence, that means interaction with the exportin. Those are typically localization sequence. That's what we mean by localization sequence. But it's not only a matter of regulation of transportation across the nuclear pods, It will be also a matter of localization for the proteins that have to be imported to the mitochondria. So the localization signal sequence of import to the mitochondria is like the address of the proteins that have to be transported to the mitochondria, to the outer membrane of the mitochondria, to the intermembrane space of the mitochondria, to the inner space of the mitochondria, and some others to the matrix. Speaking about glycolysis, they remain in the cytosol, no transportation. Speaking about Krebs cycle, citric acid cycle, it takes place in the matrix. All the enzymes involved in this process translate the mRNA for those nuclear RNA translated in the cytosol and transported up to the matrix. Okay. So this one is one example of transport to the mitochondria. If it is with plant cells, it will be also for the plastids, and that's not exactly our business by now. And that's the import to the peroxisome. All those in blue are translated by free ribosomes. So the free ribosomes will take care of the translation of all the proteins that which final localization will be in the cytosol. That makes sense. But also the nuclear one, but also the one that end up in the mitochondria and that end up in the lysosome, no, the lysosome, the peroxisome. Okay, those are the blue ones. Here they are. What's the difference in between the red and the gray? Those ones can be transported maybe only on one direction and remain stable in the nucleus, or can be transported from the nucleus back to the cytoplasm. Usually the others are transported only in one direction from the cytosol to their final destination. That's one issue. And the second one, mostly when it comes to mitochondria, you need the unfolded protein. So they have to remain unfolded for this translocation through transporters as the single chain and not the folded protein. While when it comes to transportation across the nuclear pods, because of the dimension of the nuclear pods and the characteristic we fully discussed, the transportation doesn't need the unfolding. They are transported as full 3D protein in their folded structure. Okay, they can be activated or not activated. That's a matter of regulation, but with respect to the folding, they are completely folded. Okay, when we spoke about RAN, when we spoke about the importins, when we spoke about the exportins, when we spoke about the cargo, the ribosomal subunits, it's full of those proteins that are completely folded. Okay, so that's the difference. And what the green part? The green part will be the ribosomes that will start their translation process in the cytosol, but due to a sequence which is an import to the ER, and this is at the N terminal or not that far from the N terminal part. So that means at the beginning of the translation of the protein, when the translation process takes place, as soon as those few amino acids came out from the ribosome, okay, you remember the previous slides, okay, with all the machinery of the ribosomes, and here are the first amino acid coming out from the ribosome. This one as a target, oh, we have to go to the ER. And that means it will be recognized by a signal recognition particle, SRP, and SRP is a ribonucleic particle, means it contains proteins and RNA. You remember the 7S RNA type I added on the slide and told you, keep that in mind, that one. We will speak about that one when it comes to targeting of the ribosomes to the nuclear envelope. So in the SRP, there is also this RNA. And when the SRP bind to the signal sequence of input to the ER, if it is free ribosomes in the cytosol, due to the presence of this this small nuclear RNA in the SRP, it will block translation. So translation process is blocked when the ribosome is free because it's translating a protein which has the address, the signal peptide, which is import to the ER. So can you figure out how we can remove this inhibition? You've got the point? Here is a signal sequence. Is it the SRP? The SRP inside as an RNA that will block the process. We cannot go ahead with translation. When will this inhibition be removed? When the ribosome will interact with the membrane of the ER. The interaction will be with the receptor for the SRP. So the SRP at this point interact with its receptor, which is on the membrane of the ER, and brings the ribosome close to what we call a translocon. The translocon is a transmembrane protein that will form a water channel. And now the protein can go through. I can use that one and leave you. I promised you I will leave you, so I have to do it, but it will be like this. I am the ribosome, okay? This one was the N-terminal of the protein coming out. And here there was the signal sequence to the ER. It was a free ribosome starting translation of this mRNA. Because of this signal, this one is recognized by the SRP. The SRP stopped translation. I cannot go ahead with the translation. Allow the interaction with the membrane of the ER. Interact with its receptor, doesn't interact anymore with the ribosome. The ribosome can go ahead with translation. We are on the top of the translocons, and as soon as the proteins come out, okay, it enters the translocons. I am a ribosome associated to the membrane. On the cytosolic part, no translation in the ER. Translation is always on the cytosolic part. It's a matter of adhesion to the membrane of the ER on the cytosolic leaflet. On the top of a translocons, which is a water channel, allowing for hydrophilic amino acids to go through. What happens if here I have a hydrophobic region? When it arrives at the level of the bilayer, it has much affinity for the bilayer, remain in a bilayer. We go ahead with translation. This part, on which side is it? The same of the ribosome, it is cytosolic. What's the other one? It's facing the lumen of the endoplasmic reticulum. That's the case of a transmembrane protein. If there was no hydrophobic region, all the translation process would have go with the protein going through the channel, and this one would have been completely inside the lumen of the endoplasmic reticulum. This one could be a secreted protein. Here, Golgi, vesicles, extracellular domain. This one would be secreted. What about the one that remain associated to, okay, but about the one that remain transmembrane one? Folding is not going properly. I have to unfold the protein. Okay. We mentioned this one was the N terminal at the beginning. So now it's facing the lumen. Okay. Let's say this one is a transmembrane domain. That's right. Okay. This one is a transmembrane domain. This one is on the cytosolic part. This is a transmembrane domain, a transmembrane protein. If it is one that will end up at the plasma membrane, where will be the extracellular that will be the one. This one is cytosolic. For keeping in mind orientation. I answer you right away, but I just want to stop this one. Okay. I have to stop the record.